next hour or so, I'm going to tell you a bit about the work we do in the lab, concentrating on the stuff that's related to beer yeast. I have to warn you that much of my presentation is pretty much the same as the presentation I will give to more an expert audience. I know that many in the audience are experts, others aren't. I'll try to, make it as understandable as possible, but it's a bit sort of, shipping through, finding a common middle ground. We'll see where we end up. If there's pressing questions, actually just shout them. You don't have to wait until the end. We'll see how mad it gets, but. All right, this is my lab. About half of those, the ones with the sort of sad faces, they use yeast as a model to study genetics and genomics. Yeast is a same cell as human cells, so lots of what we know about how cells work has been studied in yeast. And as a result, yeast is probably the organism that is best known from a biological point of view of all organisms. It was also the first human-like cell to be completely sequenced. We've knocked out every single gene that is not deadly in the yeast genome. So that's what they do. And the other half, the ones with the happy face, they also study yeast, but they try to make better yeast for chocolate, beer, wine, and biofuels. The ones with biofuels, they sometimes drink a little bit, so that's why they're reasonably happy, but it doesn't taste as good. That's what I'm talking about. These are the yeast cells. So these little cells here pretty much eat sugar, converted into alcohol and CO2, the bubbles, and also they make lots of aroma compounds. So they are really important for the flavor of your beer and your wine. And this is what I'm going to talk about. One thing we started with is trying to understand where the yeasts that are used today are coming from. Brewers, winemakers are all using yeast. Sometimes they buy it from companies. Sometimes they just have it. They don't always know where it comes from. They might have stolen it from the brewery next door. But they don't know. Nobody really knows, what's the history, what's the family tree, how related are they to the yeast that you could just find in nature, or how different are they? So that's the first thing I want to talk about. And of course, we then characterize these yeast, and that enables us to tell brewers or winemakers, use this yeast because this has the properties you want, maybe more than the one you're already using. One step further is to breed better yeast. We can use genetic modification for this, but we can also use sexual reproduction. So we can do exactly the same what farmers have been doing with crops and livestock, except we do it with yeast, and I'll give you a few examples. Unfortunately, many of these projects here are together with brewers, and they pay us to do the research and then get a better yeast, but they don't like me giving away all the secrets, so I can give you a few examples, but not maybe even the coolest ones. I'll tell you how we're moving on to start using computer chips to breed better yeasts. And then we'll see how far we get, but I'll try to answer the burning question, why do yeast cells produce aroma compounds? I don't know, might be because God created them that way, or not. And then, hopefully we won't get to it. It's not going to be great for the atmosphere. But I do want to, it's sort of a recent result in the lab, and we'll see. Probably it's already too late, but maybe, by the bar or so, while drinking a cocktail, I'll tell you why you shouldn't drink cocktails. All right, so I already told you this, but you might not know this. Pretty much every old brewery, Carlsberg, all the ones, the traditional ones in Belgium, have their own yeast. Carlsberg is, of course, famous, right? We're right here in the home of the city or the brewery where the first pure culture of yeast was isolated. Before then, late 1800s, before then, brewers were just reusing yeast without really knowing what it was. And so nowadays, all these breweries have their own yeasts. Even winemakers often have different yeast. They do buy it more. Brewers have it, and they make it themselves. Winemakers often buy it, but also there's many different yeast there that they buy from companies, and also those companies don't know where it comes from. And the same is true for bakers. So in our lab in Leuven in Belgium, we have this huge historical collection of yeast, especially beer yeast. It used to be really a beer lab. We had even a diploma in beer brewing, not anymore. Now it's sort of, there's a few courses still in beer brewing in the biological engineering department. We don't give a diploma in beer brewing anymore. We used to have it. And because of that, I guess, we have pretty much all the yeast of the traditional Belgian breweries, and in the meantime also gathered some from all over the world. And apart from beer, we also got some baker's yeast, some wine yeast, sake yeast, whatever, and we figured, okay, let's sequence their genomes. We're now at the point that it's cheap enough to sequence the complete DNA of at least a few hundred of them and make the ultimate family tree and hopefully learn something about the history of these yeasts. So that's what we did, and we actually teamed up with a team of White Labs, and some people, Truls is here tonight, who, well, we met at a conference in the US and we found out that, we're doing a bit the same. They had more of a focus on the American yeast, and we were focusing more on the European yeast, so we pooled our results, and so they did about half and we did half. And we got this family tree of the pure ale yeast. So these are the yeast that make specialty beers, but I'll tell you later about the Pils type yeast. Okay, so that's the family tree. We made it a circle because it's, you know, the better way to represent it. What can we see here? Well, first of all, it splits up in groups. You see the colors here? There's green yeasts and more green yeast. These are the beer yeasts. So the beer yeasts fall apart in two different groups, which we, imaginative as we are in Belgium, called Group 1 and Group 2. And then you have the wine yeast here. There's a few invaders, but mostly wine yeast. There's a clade of sake yeast here, and then the mixed clade contains a lot of baker's yeast, some beer yeast, so it's literally mixed. So that's the first lesson. These industrial yeasts are splitting up according to industry, and they are completely isolated. They're really a subfamily of the wild yeast. So it's not like you can catch a yeast from the air and think you'll make exactly the same beer as that you would make with a real industrial beer yeast. So there's something different. They've genetically diverged from these wilder yeasts. We could also put some dates on important, say, nodes in the family tree, and the oldest node that we could dates back to 1575, give or take a few years. That's really, as it looks like, where the common ancestor of this oldest and biggest beer clade started emerging, 1575, which is around the time that brewing became big business, if you will. It became more industrialized. It used to be that beer was the common thing to drink, like water. Every village had several breweries. Actually, many people were brewing at home. But around this time, you really started seeing bigger breweries. You also find records how big breweries, and that was probably key to the story I'm telling, were exchanging their yeast and sort of starting new breweries. For example, I've been to Lisbon in a brewery, and there you could actually find records where they said, okay, we actually got the yeast came with the German master brewer that helped us set up the brewery. So it kind of makes sense that around this time, yeast were really spreading. Of course, this was before brewers knew what yeast was, but they did know how to make beer. So they did know that at the end of the fermentation, you have a sediment at the bottom of your fermenter, the yeast cells, which they didn't know, and they learned that they needed to take part of this sediment and put it into a next batch of beer, thereby taking the yeast cells and literally inoculating them. So they did all the right things, except they didn't know that, you know, they were doing this with microbes and what they were doing exactly. You don't need to do this. Or you don't need to do it, you don't need to know how to do it right. And that's exactly what was happening. It was only with Pasteur and then Carlsberg that people started realizing what was going on. So it kind of makes sense that it dates back to this, although beer brewing is of course much older, but the yeast really seems to be getting isolated from their natural habitat around that time and passed around. Wine yeasts are a bit more recent. And as you can see, they're also genetically less diverse. The branches of the family tree are not as long. And we think there's several reasons for this, but the main reason might be that wine is only made once a year when the grapes are ready. And so when the grapes are ready, you make juice, you put in yeast, and then the yeast is happy, it has sugar, it can make new yeast cells, it divides, and that's when it evolves, right? You need to make children to get evolution, offspring. But of course, wine yeast can only do this for a few weeks, and then they're left to fend for themselves. They have to survive. They probably form spores and survive in and around the wineries. Whereas beer yeasts are way more happy, because beer is made all through the year, and so literally from one batch of fermentation, you inoculate the next batch, and so the yeast can start or can keep growing, undergoes many more divisions, many more generations per year, and therefore it evolved probably more quickly. Another thing that seems to confirm these estimations was that The UK yeasts and the US yeasts here, so these are the UK, these are the US, split off around 1625, time of early colonization of the US, and you imagine that some of these UK settlers took some beer yeast with them, thinking, you know, that might be handy to have. You know, what do you take? Your wife, your children, and some beer yeast, I guess. So in that sense, it seemed to confirm the story, but I totally agree with you that it is a tricky business, really predicting this right. It's quite complex, so what you do to get these dates is you need to know every time the yeast cells divide and sort of make a next generation, on average, how much does the DNA change? How many mutations do you get? And then you just count. And we have some ideas of how that goes, but as you might imagine, since it's quite a few generations, thousands, the error becomes relatively big. All right, so apart from making the family tree, we also did a lot of measurements. We pretty much made beer and wine with all these strains, but on very small scale, and we measured the aromas, we measured how much heat or cold the yeast can take, how much they're resistant to different stresses. We tried to sort of get to know them. And part of that data is summarized in this colorful map here called the heat map. Red means high, blue means low. Here you have all the yeast, so every column is a different yeast, and every row is a different property. It can be growth at a certain temperature or production of aroma compound X. We have, of course, all the numbers, but this heat map gives you clearly more information about all the yeast, or it's more visual. What can you say? Well, that you do find some clusters. For example, here, the wine yeast are clearly clustered here. One of these properties is probably alcohol tolerance. The wine yeast, on average, are more alcohol tolerant than the beer yeast, for example. Here, you have beer yeast, so again, lower in alcohol tolerance, but they might be higher in certain aroma compounds or so, whatever. So you do see some clustering, but also you see that it's a complete mess. It's all over the place. And one thing we've learned is how different these yeasts are. It's quite amazing, and I'll give you a few examples of that. One example is this here. This is the production of an aroma compound called isoamyl acetate. This is what makes banana smell of banana. Yeast makes this as well. It's exactly the same chemical compound. And later in the talk, if we get there, I'll tell you why they make it. But now it suffices to say that these are 300 different yeasts, and you see it more than 34-fold change in how much of this one compound they make. That's a lot different, right? I challenge you to think about a property in humans that ranges over a 34-fold range. IQ, maybe. Not really, to be honest, although looking at some of the students, you would think it's probably the case. Body length, no. So, you know, it's a huge variation, and these are not really outliers. It's a continuum. And this is true to some extent for many of the other properties that we tested. So next question is, can we connect the two? We got to know the genomes of the yeast, and we know the properties. Can we sometimes at least connect the DNA to the properties and sort of predict, you know, which parts of the DNA are responsible? And we've done this for quite a few, but I'll give you a few examples. And one of the most interesting ones is the following. We found that beer yeasts, looking at them from a genetics perspective, from a scientific perspective, the DNA is a mess. It's really, I call it an anti-drinking campaign. It's the first anti-drinking campaign. The bigger one will come at the end of the talk. Why is it a mess? Well, like humans, beer yeasts have chromosomes. Humans have 2 times 23, 23 from the father, 23 from the mother. Beer yeasts have 2 times 16, normally. Except beer yeasts, for the first chromosome, they might have 3 copies. For the second chromosome, they might have 5 copies. The third chromosome, they might have 2 copies. For the fourth chromosome, they might have part of it, 5 copies, and another part, only 3 copies, right? So these chromosomal changes in copy number of chromosomes in humans are highly deadly. There's only a few that we can survive. The most known one is trisomy 21. When you have 3 copies of chromosome 21, you get Down syndrome. Most of the other ones are deadly. They cause early natural abortion. In yeast, this is not the case. It's crazy. All of these guys have several of these abnormalities. And when you start looking at what genes are on these chromosomes that have been duplicated, it looks like it's not random. One example is that beer yeasts on average are much better, don't even look at the graphs, just listen to the words. Beer yeasts are much better at eating maltose. Maltose is a specific sugar that is very much present in beer medium, but not so much in nature, and definitely, for example, not in wine medium. So clearly these beer yeasts have learned to eat the sugar. And if you look at the genes that are responsible for eating the sugar, they are in these parts of chromosomes that have been highly duplicated. So the yeast just have extra copies of these genes which allow them to grow faster and eat more of the sugar. And there's other examples of this. Another thing, and that is I'm not looking at chromosomes anymore, but now really at the letters of the DNA, we do find very specific changes in the DNA code for beer yeast. And one example is a cool one. Beer yeasts apparently have been selected to have errors in two genes called PAT1 and FDC1. And without going into the details, these two genes encode enzymes that form a flavor that you don't want in beer. The flavor is called 4VG, 4-vinyl guaiacol. Only Germans want this in Hefeweizen beer. So if you drink a German Hefeweizen, there's this spicy flavor. Clove, some people call it dentist, smoked sausage, although that might be in Germany because the sausage was too close to the beer. But that kind of flavor that you normally don't find too much in beer, you find it in German Hefeweizen. You find it in a few beers, right? For example, Leffe is a Belgian example. So in some beers it can be considered good, but in most beers it takes away from the freshness. You don't want that. And so beer yeasts, again without going into details of diagrams, clearly we can show have been specifically selected to have errors in those genes so they don't make these flavor compounds. Wine yeasts don't have these. Not that you want your wine to smell like smoked sausage, except in Germany, but the precursors of this flavor are not present in grape juice. So whether wine yeasts have these functional genes or not doesn't matter. They never have the off flavor. This, by the way, is the biggest mystery of our work yet, because when we date back these mutations, they date back to pretty much almost the origin, the 1600s, way before microbes were known. And so it seems like brewers were somehow selecting yeast to not make these flavors, because if you look at all the wild yeast, they do make it. But to select for yeast that would have these mutations would almost mean that they would have to go to single cells and make colonies and literally do microbiology like we're doing in the lab now. And we don't really believe that's the case, but on the other hand, we don't know how else it could have happened, unless not having these genes gives you an advantage in beer, but that also doesn't seem to be the case. So to be honest, we don't know. We just drink another beer, we go like, ah. Conclusion from this part, beer yeasts are dogs and wine yeasts are cats. Beer yeasts have been really adapted to the living in the brewery. They've been completely domesticated. They've adapted to this human-made environment, but if you would take them in the wild, they wouldn't do very well. They've lost a lot of stress resistance. I told you they're less alcohol resistant because beer usually, except maybe if it's not very strong, 5, 6, 7%, not like 13, 40% like wine or even higher. But then they're very good at eating the sugars that they don't find so much in nature, but a lot in the breweries. Wine yeasts are a bit wilder because only 3 weeks they're in heaven, and then the rest they're more in the wild, and you can see that they intermingle more with wild yeast as well. How can we make yeast even better? Well, one way is genetic modification, and I don't want to talk too much about this. We do this quite a lot in the lab, but mostly to investigate how these yeasts are working or which genes are important. Not so much to give genetically modified yeast to brewers. Most brewers do not want them. Beer is a quite traditional product and they want to keep it that way. Also in the marketing, consumers wouldn't necessarily accept this, or at least not all consumers. So definitely the big brewers don't want to have anything to do with it. We do get some questions from the US because they know we're doing this, and some smaller craft brewers are asking us to send GM yeast. They want to use this because they figure, well, at least some people will drink it because it is GM and it's cool. Others might not, that's fine. We don't give our yeast to them because we're not sure that they would follow all the rules and get approval for these yeast. Not because we think the yeasts are dangerous. We do drink some of these beers. It might change with the coming of CRISPR-Cas. I can tell you afterwards, it gets a bit technical. But so maybe in the future, we'll get to see more of these things. What can you do with GM? You can, for example, make yeast cells stick together. Here you see a test tube with yeast cells in suspension, as they normally are, so they're individual cells, and that's why the medium is turbid or cloudy. But when you activate one gene, or overactivate it using GM, you can make them stick together. And brewers like this because at the end of the fermentation, the yeast needs to sediment. Same in champagne, the yeast needs to sediment in the, they put the bottles upside down, the yeast needs to sediment in the neck of the bottle so they can freeze it and shoot it out, or brewers, they just, pour off the beer. So you want this to happen and to be as strong as possible at the end of fermentation. With genetic modification, you can do this, no problem. You can take this isoamyl acetate, this banana flavor, you can overactivate the gene for this, and you can make 200 times more of this flavor compound and have beer that smells like banana milkshake. Not very good, but it's cool. So we're not using these techniques for brewing. What we are doing is breeding, male and female, a candle and Barry Manilow, and let them do what they do, and a few hours later you might already have a hybrid, and then you can repeat this. Or another way is experimental evolution. This is the weird thing about yeast. It has sexual reproduction, where you need A and alphas, or males and females. I'm not going to explain sexual reproduction here, not yet. But they can also do asexual reproduction, like bacteria. So they can literally make clones of themselves. But even when they do, they're not perfect copies. Every time the DNA is replicated, there's a few errors happening, a few mutations. So when you grow yeast asexually, even then you get changes in the DNA. And if you drop them into conditions that select for something, for example, you drop them into high alcohol concentrations or in very low temperature, you might have a few cells that are more resistant to this. They start to divide more quickly, and as the process continues, you get better yeast that are better adapted to your environment. So we're just abusing Darwin's theory here for our purposes. One example we've taken some of the most flavorful fruity yeasts, bred them together, and made yeast. The parents here are the colored dots. This is the best parent always. And some of the open circles are some of the offspring, and we created yeast that produces 50% more, so around here, of even these best parents. And this is natural. These yeast you can immediately use. They're like, crossing tomatoes, if you will. So no problem. We sold this yeast to a Canadian brewery who wanted to make a beer that was only 6% in alcohol, but would have the aroma of a Belgian triple, so quite fruity and strong. We've done, and actually I should say Tim, who's also here in the audience, has spent a lot of work on making yeast that are more alcohol resistant, for biofuels, of course, but also to make stronger beers, which you can then dilute with water. I don't know if you know this, but most of the, especially Pilsner beers, are made to 8%, even 9% of alcohol, and then just before they're put in the bottle, water is added, which sounds really wrong to do, but it's actually not bad. The beer tastes good, and it is good for the brewers because they can make more beer in a smaller brewery, and it is good for the environment because you need less energy and cleaning products to make this larger volume of beer. So that's something we've done. An example here of some commercial biofuel yeasts increasing alcohol concentration, and you can see that some of these hybrids really grow at higher concentrations. And this is another example where we start a fermentation, a biofuel fermentation with lots of sugar, 350 grams per liter of sugar, so it's almost like syrup. And you see how the yeast cells over time start eating the sugar, and of course making alcohol, that would be going up. You see that the commercial yeast cannot finish all the sugar, it's like too much alcohol, they stop. But some of our hybrids do it faster and finish. And there's a funny story to this. Tim actually made, I don't know how many thousands or millions of crosses, Tim, right? It was quite some work. But we also had one not so very talented master's thesis student in the lab who made, I think in 6 months or so, he made something like 10 crosses, which, trust me, that's what Tim does in a second. But one of those freaking 10 crosses was literally the best strain we have. So somebody questioned me whether this was the least talented or the most talented student I've ever had. Lucky, yeah. I invited him for a trip to Las Vegas. He didn't understand why. And now we're also combining the knowledge we have on the DNA sequence with the breeding. We can, in some cases where we know which parts of DNA are important, we can pick those parents that have this exactly what we want and breed those together. And instead of testing the hybrids or the offspring, we're now doing a small DNA sequencing to see if they have the right pieces of DNA and combinations of DNA, which is something they're doing in the plant world as well. You might imagine if you want to breed trees, for example, it takes a while before a small tree becomes fertile, so it's great if you can do this. For yeast, the gain is not as big, but still it helps us to create better yeast. One example of the experimental evolution that I was talking about, remember this is Darwin, work by Karin, where she grew yeast for 5 years in a row, just every day they were growing in what many people might think is heaven, high alcohol. So we're just feeding them alcohol all day, and as soon as they sort of got better, like, okay, we're feeling comfortable in 6% alcohol, we increase the alcohol to give them again stress and to again try to get offspring that was even more adapted. And this works. One of the cool things you can do then is you can walk back in evolution, and that's something that's very difficult. We have fossils that was a cornerstone of Darwin's theory. What we don't have is really ancient DNA. We have some frozen samples, but that's 10,000 years ago. That's nothing on an evolutionary timescale, especially not for bigger animals. Here, fine, it's only 5 years, but yeast is reproducing every 2 hours, so it goes quite quickly. And we can take samples every 2 weeks and know exactly when we've taken them and sequence them, and we can literally see how the DNA is changing. And so we've done this. And what you then see, this is the population of yeast from 0 to 100%. You can see around, this time here, you have the purple mutation in a gene called PDE2 that's clearly increasing the alcohol resistance of the strain. And therefore, this population, this mutation is spreading in the population. It started with one yeast who had this, but because it had this mutation, it could generate more children than yeast that didn't have it. So slowly this mutation spreads, and that's what you see here. And then it stops, probably because there's other mutations that occurred here, but at lower frequencies and many different ones that start competing. So what used to be a really good strain here is now not better than the rest anymore, but the rest is very mixed, so we don't see all those mutations, or we don't represent them. And then suddenly you got this red mutation here in 2 genes actually, that very quickly spread through the population, very strong mutation, and then in that background you get extra mutation and extra, and then here you have 2 competing ones, and in the end the green ones win. And we've done this for all the 6 populations that we evolved. So that was quite cool. We could walk back, but we could literally also prove that some of these genes were really important, and if we put some of these changes in those genes into biofuel strains, biofuel yeast, we could make them more alcohol resistant, and we've patented those so that we can use those and no one else. Well, they can if they talk to us and send us some alcohol, maybe. Lager yeasts. I don't know if you know these. Pilsner beers, Carlsberg, is made or are made with different yeast. It's a very different kind of yeast. It's actually a cross between 2 different species. So here you have an example. This is a liger. It's a lion crossed with a tiger. This is not photoshopped. It exists. It doesn't exist in nature. It only exists when you have 2 frustrated animals in a zoo. It also doesn't exist in nature because lions and tigers don't share the same habitat. A zonkey, a wolfen, this is all true. And this is the lager yeast. It's arguably less impressive, but way more useful because it makes our Pilsner beers. So we've started sequencing these genomes, and they're very special genomes because you literally have part of DNA coming from one species, and one species is actually the normal ale yeast, so Saccharomyces cerevisiae, the pure species that I was talking about before. The other yeast, called Saccharomyces eubayanus, is a cold-tolerant yeast. It grows quite well in cooler temperatures, say 10 degrees centigrade, where the normal ale yeast doesn't grow at all, or almost doesn't grow. So we've sequenced them, and then for the Saccharomyces genome, we can put it on this family tree. So this is the same tree that I already showed. It looks a bit different, but it's the same tree. The wine yeast here, the beer 2 and the beer 1 clade, and you see that the two parental strain, the two lager, back off Verstrepen, easy. The lager yeast split up in two families, which are called Saaz and Froberg. This was already known before we did our sequencing. We've sequenced a bit more, and we can really show that the cerevisiae part, so the ale yeast part of the genome, clusters with beer clade 1, so the oldest beer clade, and then specifically with the German part of it, which makes sense because this likely happened in Germany when they installed the Reinheitsgebot. The Reinheitsgebot are a set of laws, medieval laws, around 1650, 1700s, I think, where brewers were forced to start brewing beer. No, it's actually earlier than that. When brewers were forced to start brewing beer in their basements, also in winter at cooler temperatures, where our normal beer yeast couldn't do so well. But then probably spontaneously some hybridization occurred, and you got now this hybrid, and we've reproduced it in the lab, that could make great beer and eat lots of sugar and produce alcohol, because this cold-tolerant one, this eubayanus, is not so good in making alcohol. But it is good at growing at lower temperatures. And so that's another reason why we think our dating sort of matches, because this again also matches with that. Once we know what happened there, we can make more of this. And this, by the way, is also the yeast that was isolated right here in Denmark for the first time. The Unterhefen number 1 is one of these special hybrids. Once you know how this lager yeast comes about, you can make more of them. There's only 2 natural families, and that also explains why there's not so much taste diversity in lager beers. The recipe is pretty much set, and then the yeasts are so closely related that they're not giving you that many different flavors. So one student in the lab took different cerevisiae and different eubayanuses and started crossing them, and then made beers with them. And we profiled we measured 50 aromas to get sort of a mathematical diversity in aroma of these beers, and this is represented in this 3D graph. The green and the red ones are beers made with the traditional existing lager yeasts, and the blue ones are beers made with our new yeast, and you can see that we expand the aroma diversity. I must admit that some of these beers sucked like crazy, but some of them are quite nice, and some of the strains are now used in production. The future lab-on-chip approaches. Breeding, before we had Tim, who was breeding, then after Tim left, we figured, let's buy a robot to do this. Because the robot will do even more work in a shorter time, and doesn't eat as much as Tim. No, Tim also worked with this robot. It allows us to make 10,000s of crosses. The problem is not making hybrids. With yeast, this is easy. But testing them, right? How do you know which are the good ones? Certain things you can easily test, like alcohol resistance. You dump 20 million yeast cells in high alcohol, and if 5 survive, you know which are the good ones. And nobody cares about yeast cells, so we don't have protesters in front of our lab that we're doing cruelty on yeast, so that's fine. But for aroma, you cannot do this. You have to make beer and measure the aromas. And that's a tricky part, or to know how quickly they can eat sugars, for example. It's not so trivial. So we used to do this in this kind of scale. We can do it for hundreds at a time, but not for thousands. So we scaled everything down to 2 centimeters by 2 centimeters. I should actually pull it out of let me pull it out of my backpack. Normally I do the Apple trick where I have it in my backpack. You've seen the introduction of the Airbook, yeah, where Steve Jobs pulls out the Airbook out of this brown envelope. I normally pull this out of my pocket and I say, ooh, I have like 10,000 breweries here. But that effect is gone now. It's the cocktails. But so, ooh, I have 10,000 breweries here. Thank you for that reaction. So this is how they look from the inside, these 10,000 breweries. Well, they're not breweries, they're fermenters, to be really honest, but breweries sounds cooler. You have small droplets with beer medium in which we can inject one yeast cell. And you'll see, you know, it's quite simple in principle. You just inject beer medium into a specific kind of oil. You'll see it zoomed to here soon. And then you get these stable droplets. Getting the right oil was tricky, but we managed it. So yeah, that's how they're made. And then here you can actually, it's a close-up, so you can see the yeast cells sitting in there. There's more than one now because they can grow in there. So there's like 20 or 30 or so in there. They multiply when you feed them. So we literally have mini fermenters now. We need to measure stuff in them. So we copied this device from a lab at Harvard where we can merge droplets. So this is the first droplet that gets stopped, and then you can intersperse these droplets with droplets that contain enzymes, so you can measure stuff. So you can add some color reagents that will color the more sugars are left or whatever, and then you can start measuring things in these droplets. And then we need to sort the droplets. This we do in two ways, but the coolest way is this one, only for the name. This is with a microjet heater. So what this is a combination of small galvanic elements. So these are pieces of metal that can heat up or cool down very quickly. And you see that they have these fins on them. So they have a lot of surface. When they heat up, there's a small steam layer that is right here on the surface, and it creates a pressure difference that can push a droplet down if that's the one you want, for example, because this yeast is eating more sugars than the rest. So it's not working perfectly yet, but we're getting further and further. We actually now have some quite cool results, but they would be quite boring to show. So soon, hopefully, we'll be able to speed up the breeding even more. Why do yeast cells produce aromas? I kind of realize I'm the only thing between you and more cocktails, so I start feeling uncomfortable. So I'll speed up a bit. Why do yeast cells produce these flavor compounds? These are a few of these compounds that yeast cells make. This is one specific category of flavor compounds called esters. They're volatile, and they're the same compounds that you also find in nature. I already talked about the banana flavor. But here you have pear, here you have roses. I don't know, do you know rose water? You can buy it at the pharmacy. It doesn't really smell like roses, but a little bit. That's this compound, isobutyl acetate, raspberries. So yeast cells all make these. Question is why? And this is something that I and also Sophie, who's also in the audience, tried to solve during our PhDs. The thing is, you can knock out the genes that make these aroma compounds, and you can do everything you can with these yeasts, and they grow just fine. They don't seem to miss these genes. They just don't smell good anymore. But, you know. Until on a Friday night, I was eager to get to the bar. This really happened. And I left my flasks on my bench. So I had a flask with one yeast that produced, that's the banana milkshake yeast, the one that produced 200 times more of this flavor. And that's one that is just normal, and then these yeast don't produce any esters. And we have a fruit fly lab next door. Fruit flies are used as a model in neurobiology. That's one thing that yeast are not so great for. They don't have neurons, so studying the brain, for example, in yeast is a bit difficult. The problem with these fruit fly labs, I don't know how many scientists there are or microbiologists in the audience, but if you ever lived or try to do experiments next to a fruit fly lab, these goddamn fruit fly scientists cannot keep their goddamn fruit flies in their goddamn labs, okay? It's really annoying. It's not like we ever go there and sort of pour microbes. But they find it normal that their fruit flies are all over the place. But in this case, it was useful because 50 of them found their way into our very fruity yeast, and only 2 of them crawled into this, and to be honest, to make this a perfect experiment, this guy would probably have to be there because maybe these guys just missed their landing and sort of crawled in the wrong one. But of course, it made us think, well, you didn't have to be Einstein to figure out, like, wait a minute, you know, fruit flies like fruit, and maybe they don't go by their eyes, but maybe by their noses or their antennae. So many years later, I finally was able to gather funding for this project and hooked up with a neurobiologist, Emre Aksi, one of these annoying fruit fly guys, luckily in a different building, so I was okay with him. And he was studying neurobiology in flies, and he has these arenas. That's another thing about fruit fly scientists. They exaggerate. The arenas are about this big. And you can flow air from the four corners, or you can bubble that air through beer, so it picks up the aroma of the beer, and then you can study the fly behavior. And in the beginning, we're just flowing air from the four corners, and you see that the flies don't care. They like corners for some reason, so they go a lot to corners. But at this point, we're starting to blow air through normal beer, and then a beer made with yeast that doesn't produce the fruity flavors, and just air. And you can see that clearly the yeast like this, the flies like this corner. And then we go back to air, and they disperse again. And you can measure this. This is the fly density in the four corners. So 2 gray lines is just air. You have the normal beer and then the beer with the not so fruity yeast. In the beginning it's all air, so the flies don't care. Then they go clearly to the nice smelling beer aroma corner. They don't care about this mutant here, or beer made with a mutant that doesn't produce fruity flavors. And then you can start adding, one by one, you can start adding the aroma compounds back and see what the flies are reacting to. So here we added this phenylethyl acetate, so that's the rose's aroma. And guess what? Fruit flies do not care about flowers. That's why they're called fruit flies. Important conclusion of our paper. They didn't care about the stuff. But then when we started adding banana flavor, and when we started pear and banana, here you get completely the response as if it was a normal beer, although it lacked some other flavor compounds. Pear and banana together, that's what the fruit flies wanted. Emery, being a sadistic neurobiologist, wanted to go a step further, so he took some of these genetically modified fruit flies that have a specific genetically modified version of a calcium receptor in their genomes that made their brains light up in green fluorescent signals whenever a neuron is firing. The neuron lights up. And so what you need to do is you need to immobilize this fruit. This is not a joke, right? You need to immobilize this fruit fly under the microscope, slice open their skulls. It takes kind of a steady hand. You cannot hit the brain, mind you, because then they tend to die. But just slice open the skull. It's a bit Hannibal Lecter. And then you can actually see the neurons as they're firing. And to add insult to injury, you then expose them to beer aroma. So you make these poor animals think that, beer. And so these are brain scans of fruit flies exposed to various kinds of beers. So this is probably also how my brain would look like when you let me sniff a beer. It fires up. This is the response to a beer made with a yeast cell that doesn't produce the fruity aromas. It's just one gene that we needed to remove. So yeah, clearly not so much happening, because this is the aroma. Where is it? The aroma to paraffin oil. Yeah, that's completely blank. So here you see something. There's some alcohol in there, vinegar, but not so much. And then you can add again the different flavor compounds. And then when you add banana and pear, you again get a very strong response, very similar to the normal beer. So clearly, yeast cells are attracting fruit flies with the aromas they produce. Why? Because they can hitch a ride. These are fluorescent yeast cells on the not so sexy leg of a fruit fly. So as the fruit flies come, they actually eat the yeast. For them it's a protein meal. It's something I didn't know. There's many things I did. For fun detail, we had to work with, virgin female flies. This is not because Emery is kind of a psycho. Well, maybe he is. But it has something to do when you start mixing boys and girls together. You get complex, maybe not so complex behaviors that destroy your results, right? Anyway, what we also learned, or what I also learned about fruit flies, is that they have to have a protein meal before they lay eggs, the females. Just like mosquitoes come and suck your blood to get proteins. It's only the female mosquitoes, of course, that do this. Because they have to do the offspring. I didn't mean anything bad. The same for fruit flies. They need to get a protein meal. They don't get it from drawing blood. They get it from eating yeast. So for them, fruity yeast is kind of attractive. What's in it for the yeast is that as the fruit flies are eating the yeast, some of them get caught on the body, even way more on the face. And so that's one way to travel. And it's kind of an expensive taxi if you have to sort of offer up your brothers and sister to, you know, get a ride. But it kind of makes sense, because if you're on a grape and the grape is almost out of sugar, you want to go to the next grape. And trust me, as a yeast without any insects around, that's a difficult thing. You don't have wings, you don't have legs, you don't even have a lousy flagellum. There's just no way to get there. Except for to offer up a few brothers and sisters, but that's okay because there's a few millions of them anyway. And you might get to the next grape, and the fruit flies are going exactly where you want to be. Fruit, sugar, that's exactly what yeast wants to eat. So I think it's really, or we believe it's really a nice collaboration between the microbes and the insects. And then the last part, are you sure people that want to leave the room, this is a chance. Actually, this should be read now. Why you shouldn't drink. Remember the experiment of Karen, where she was growing yeast for 5 years in higher and higher alcohol concentrations? We were trying to make more alcohol-resistant yeast. We're also doing this with students, by the way. So she got quite crazy, because as she started sequencing the genomes, these yeast didn't have 5 mutations, so changes in the DNA. They had hundreds. Well, or definitely many tenths. About an order of magnitude more than what we expected. And that made her life very difficult. It made us wonder why. And of course, the first thing is, well, maybe the alcohol causes mutations, like sunlight causes mutations. It gives you cancer. And sure enough, we kind of knew this, but we didn't want to know this, a bit like, I guess, all of you. When you start looking up, is alcohol a mutagen? The first thing you find is, oh yes, and it causes a lot of cancer. You know that, I guess most of you know that cancer is a disease that is caused because of damage to the DNA, so mutations in your DNA. So anything that causes mutations, per definition, is a carcinogen. And alcohol is a class 1 carcinogen. It's just as bad as cigarettes. The strongest carcinogen category we have. What kind of cancers does it cause? To the mouth and the upper throat, the larynx, the esophagus, breast, but that's special probably because it changes the hormone levels in females. Liver, much less than I thought. I always thought about liver cancer, but that's actually not the biggest increase, and bowel. So mostly the organs that get in direct contact with the alcohol. And so people thought they knew how this, when you look up the literature, people think they know how this happens, because ethanol, when you drink it, is broken down to acetaldehyde. That's the first product. That's also blamed for the headaches you get, or part of the reason. I don't know if that is completely true either. Acetaldehyde is very reactive, and it's thought to bind to your DNA and therefore causes changes in the DNA, which can then cause cancer. It might also create reactive oxygen species. So that was the mechanism. And I'm not going to bore you with all the details. I just want to get you the basic idea here. We think the mechanism is more complex than this. When we started looking into this, we can block the formation of acetaldehyde. It doesn't do so much. We can add acetaldehyde, pure acetaldehyde, to yeast cells. With humans, it's not so ethical to do it. And you need to add 100-fold more than what we can measure inside yeast cells to get the effect the mutagenic effect. So it doesn't seem to be everything. And so then we teamed up with Michael Lisby right here in Copenhagen. After we did an RNA-seq experiment, this is, without going into details, is an experiment to see how the cells are reacting to something. So we gave the yeast cells alcohol, and then we measured how the cells responded. And one thing we found is that they had proteotoxic stress. Their proteins were under stress, probably because the proteins are not stable when there's too much alcohol. Proteins are very stable and normally created to work in an aqueous water environment. When you add alcohol, the properties of the solution in the cell change, and therefore some of the proteins start unfolding, and that's the response we saw really jumping out. And Michael told us that this is known to cause stress, also to cause problems with the replication fork. The replication fork is a big collection, a big complex of proteins that replicates your DNA. So what you see here are the 2 strands of DNA, and then here you have the 2 copies. This takes you back to your bachelor years, right? And one telltale sign for proteotoxic stress is MRC1 that's being recruited away from the replication fork. So sure enough, as I'll show you in a second, when we add ethanol, MRC1 is recruited away from the replication fork. This whole thing becomes unstable, and then something weird happens. You get other compounds here, the real replicases, which are right there, the enzymes that really copy the DNA, that normally don't make almost any errors. They're some of the most precise enzymes we have because you do not want too many errors in your DNA. They get replaced by sloppy ones, error-prone polymerases. And then they continue the copying of the DNA, but then at a higher error rate. So here again, you're bored by now. So here you just see that when you give ethanol, you see MRC1 making foci, and that really is the telltale sign for it being recruited away. And when we delete the error-prone polymerases, the mutagenic effect of ethanol, so normally 4 or 5 times more mutations, when you delete the error-prone polymerases, or at least some of them, almost the complete mutagenic effect of ethanol is gone. And we have way more data that I'm not going to bore you with. But it looks like, unfortunately, we were right to say that ethanol is a mutagenic. So I don't recommend you to drink more than 20, 25 cocktails tonight. But we were wrong about the mechanism. And one thing we can do, and we're working this on, of course, is we can try to knock out these error-prone polymerases in humans. No, that's not true. But it would be cool. Few last slides. Very recently, a few weeks ago, we officially opened what I consider to be the most fun piece of equipment in my lab, which is literally our own little brewery here. So finally, we can make our own beers at a larger scale. For lots of the industrial projects we do, just the lab scale is not good enough. We need to be able to move up because yeast doesn't always behave exactly the same in 1 liter bottles compared to 500 or 5,000 liters. And another thing that maybe for some of you might be relevant is that we'll be soon opening new positions for junior PIs. You get an incredible startup package and fun colleagues and a brewery. So shoot me an email if you might be interested. We're looking for people with really strong CVs, good publication records. It is a strong selection, but if you think it's you, just shoot me an email. You can find my email, just Google my name. It will come out. If you need to know more, these are our publications. The main publication is this one in Cell, but there's a few others. The one on the flavors and the flies was here in Cell Reports. And by the way, one last thing that gives you something to look forward to. This is my shot at the IG Nobel Prize. You know the IG Nobel Prize is sort of the Nobel Prize for silly, useless research. Not really, but a bit. We're now testing which beers are the most attractive for fruit flies. We think this is a great way to spend taxpayers' money. I need to thank all my students and postdocs because they're now hopefully, well, probably not now anymore, they're doing all the hard work. And I want to thank you for coming here and the organizers for organizing this quite cool event. It was really fun to be here. Thank you.